Deoxyribonucleic acid, or DNA, is a complex molecule that consists of many components. Let's break down the components into 4 groups: basic elements, nucleosides, nucleotides, and nucleic acids. Each of these 4 groups builds upon the other to form the familiar structure of DNA. The basic elements that compose DNA are 5 atoms: carbon, nitrogen, oxygen, phosphorus, and hydrogen. A nucleoside is the combination of these atoms into 2 structures: a 5-carbon sugar molecule called deoxyribose, which is responsible for the name of DNA, and one of 4 nitrogen bases. The 4 types of nitrogen bases found in DNA are adenine, thymine, guanine, and cytosine. Together, these 2 structures form the supporting backbone of DNA. A nucleotide consists of the previous nucleoside and a phosphate group. When nucleotides form chains, they are referred to as nucleic acids. Thus, DNA is a type of nucleic acid. In the living cell, DNA is not produced as a single strand, but on a template formed by a pre-existing DNA strand, creating a second strand of nucleotides that is attached to the template strand, thus forming a double strand of DNA. The nitrogen bases protruding from the existing strand bind to bases of the strand being synthesized according to the base pairing rules. Adenine binds to thymine, and cytosine binds to guanine. Note that the second strand is arranged in the opposite orientation or upside down relative to the existing strand. For orientation purposes, the carbon atoms in the nucleotide sugar are numbered 1 through 5. The phosphate group is attached at carbon 5 and is appropriately called the 5' end. The next nucleotide in the chain attaches at the 3rd carbon and is called the 3' end. Therefore, each nucleotide strand has a 5' end and a 3' end, and each strand is in opposite orientation to one another when in a double strand.